I'm Julie Forman-Kay, and I am a senior scientist and head of the molecular medicine program at the Hospital for Sick Children Research Institute in Toronto, and I'm also a professor in the Department of Biochemistry at the University of Toronto. I am interested in intrinsically disordered proteins, and most people may think about proteins, that's what you eat in the morning, you know, eggs and whatever, gotta fuel your day. But proteins are the primary movers and shakers in biology, and the way they are constructed is like a necklace, beads on a string of amino acids. And many people understand the way proteins function, the way they understand human machines, you know, like an engine, that you have a visible form, and that form gives rise to function. And proteins can go from this wiggly beads on a string necklace to form a rigid structure, fairly rigid, that gives function. But about a third of the beads, let's say, that make up the collection of proteins never form a stable structure, and they function in this very wiggly way, like a necklace would, making all kinds of conformations. And so I am interested in understanding how this large amount of wiggliness in biology can actually do function, and what happens when there are variants or mutations that cause disease in these regions of proteins. How do we understand how a difference in a wiggly bit of a protein can cause pathology? So I'll tell you two stories. One, we're just starting. So my graduate student, Sayantini Upadhyay, is looking at a mutant of a protein called DDX3X, which is a very important autism spectrum disorder associated gene. And this variant is associated with neurodevelopmental disorders in general and intellectual disability. And we're trying to understand differences in phase behavior just, of this disordered region that has the variant in it. But with NMR, we want to understand what the chemical interactions are in the normal protein, the wild type, and in the mutant. And so we're doing two classes of experiments. One is using a more standard approach called paramagnetic relaxation enhancement. And there you basically put an unpaired electron that lowers the signal for the NMR, and it tells you something about how far, what the distances are, and you can come up with restraints for these ensemble structure calculations that I was talking about. But the other kind of restraint we want is what is called proton-proton nuclear Overhauser effect. This is called NOE, and it's another kind of distance information. And Lewis wrote this beautiful set of experiments that are particularly valuable inside condensates. And so we're going to be applying those tools. And so that's one experiment that's one project that we're really looking at, trying to understand what's wrong with this protein. You know, I think one of the problems with autism in terms of there not being any cures is they don't understand the mechanism by which the mutations cause disease. So we're starting there. Another project that we have is trying to understand more of a fundamental biophysical impact of condensates. So how condensates change biology. And one of the things that they do is they do something called partitioning. They suck certain molecules in and exclude other molecules. And that's sort of easy to understand. But we're more interested in how they impact. Let's say you have a folded, stable structure. It may not be so stable inside the condensate. The condensate may make it more wiggly, may make it unfold, or parts of it open up. If the folded protein you're interested in is an enzyme that is really important for a particular biological process, then that could have significant impact. And then if there's a difference in this, basically the solvent environment caused by mutations in that environment, then you could have a different impact on the enzyme. And so we're studying a particular enzyme that takes RNA and chops it up a bit and is the beginning of degrading the RNA. So it's a way of controlling the proteins that are made from genes. And this enzyme is normally locked closed, has a little tail that locks the site that would enable it to do its function. And when we put it in a particular condensate environment, the tail opens up. And so we're using NMR and other approaches to try and understand how the condensate environment impacts the opening and closing of the tail. So I think in general, my work is aimed at understanding an underappreciated part of the whole biology. People don't appreciate the impact of intrinsically disordered proteins at all. They don't understand how they function or how disease variants can cause disease. And so my work is very fundamental on some level, but we are actually studying autism, we're studying cystic fibrosis, and we're studying Ewing sarcoma. We have these other projects that are all focused on condensates and disordered regions, and some of them use NMR more than others, but in the past we've certainly used NMR a lot on our studies of cystic fibrosis, and to look at interactions that we now know help drive condensate formation, clustering, which is associated with function that is not happening in cystic fibrosis disease. So I think that the ultimate impact is basically understanding molecular mechanism of disease involving disordered regions that can set a foundation for thinking about future therapeutics. And I'm not so much in the therapeutics, though I interact very closely on the Ewing sarcoma project with a clinician who's developed a small molecule that's in clinical trials for Ewing sarcoma, and we're trying to understand the mechanism of action of that compound. And I also have a collaborator on the cystic fibrosis project who's trying to come up with new therapeutic modalities to modulate the condensation or the clustering of this protein in order to have a suggested future therapeutic. So there's hopefully going to be direct implication